Now, waste is a proof of abundance, surplus, which should not create crisis. I make a second statement: there is no oxygen in air. If you do not agree, sir, please join me right and correct me right. Now, are we in the world of biotechnology or in the time of technobiology? I think it is the later, because what is biotechnology in my understanding, I'll share it. There is no industry on this. Now, let us identify problems. A pandemic is a short-term problem. Climate change is a long-term issue. But the sewage management is an eternal concern. Because if you see the 5,000 years of pandemic history, they are urban in origin, number one. And they have their origin from the sewage. So the sewage provides the excel of civilization. In all SDGs, if focus on the sewage management, scientific sewage management, a lot can be achieved. Now, a coincidence after coincidence in name of a coincidence is not a coincidence. If you see 5,000 years of pandemic history, it's not a coincidence, it is some illiteracy. So, on this forum, you hold a central position because microbes are the roundabout routes to all the metabolic and ecological pathways. When it comes to microbes, we have to deal with sewage invaders. Secondly, we'll have to understand toxins. Thirdly, what is BOD? The whole engineering world and aquatic people talking about BOD, they are on a flawed path. Now, the clue to the whole understanding comes from reverse Krebs cycle. I do not know our present textbooks mention that. स्नेहल जी कुछ बोलते हैं अपनी टेक्स्ट बुक मुझे आई एम नॉट कनेक्टेड विद रेगुलर टीचिंग
Yeah, Krebs cycle, it is there always.
नो रिवर्स
It is there. Reverse Krebs
It is
cycle
there, it
also-
is there.
वंडरफुल या नाउ वाटर सॉइल मैजिक इज अनदर एरिया जो स्नेहल जी ने रिपेरेंट जोन में कहा कि जीरो ऑर्डर स्ट्रीम इज नॉट टच्ड नाउ synergy and serendipity, that is one more area we'll have to address. And nature does not reinvent principles. Nature is very conservative. Nature is very orthodox. And this area is weapons of mass destruction which we all produce. None of can ever say that I have no role. Now, what is sanitation? What is sewage? Why understand that? All the microbiological learning has been focused on the public. Now, sewage is a climate science. Microbiologists need to address this aspect of sewage. We all know that the sewage production was not under lockdown, even for a day, anywhere on this planet. For sustainable growth, the future cities, we are in for a huge U-turn, which has no component of business as usual. All STPs world over focusing on water and nutrient recovery is hugely flawed because water recovery and nutrient recovery is totally irrelevant to sewage science. Now, let us take first point, sewage illiteracy. Do we know the price our civilization paid for this ignorance? Can anyone, not Snehalji, can anyone identify this man in white hat? Now this was for microbiologists. This is a slide I want you to focus on because in a correctly folded protein, the seventh position is taken by serine. In BMAA, which is a cyanotoxin, changes the folding of the protein molecule, an entire synaptic junction transmission and motor and neural controls are disrupted. Where do we go? Or where did we go wrong rather? Microbiologists, I am personally sharing my concerns here. They have focused on pathogens for medical health, public health, and industrial benefit for profit. Microbiologists have not addressed other areas at greater depth as compared to these two domains. And this is climate. Microbes responsible for climate stability, mainly archaea, are not culturable with present knowledge of cell and tissue culture biology. You cannot isolate any microbe, archaea, or some anaerobic fungi linked with climate stability. And they all function with the nitrous oxide and all that because denitrification and nitrogen sequestration is the domain of denitrifiers, which are archaea and fungi. Now, understanding oxygen, I made a statement that there is no oxygen in our environment, in air. How many of you would agree or disagree? कुछ बोलेंगे सब लोग तो अच्छा रहेगा. Okay, let's go. This is oxygen. And we are playing a seesaw, which is oxygen radical, which is O or OH hydroxyl. As a hydroxyl, it's a bound oxygen. That is elixir. Second form of oxygen is an ion. There is a difference between radical and ion, which is a very reactive species. That is toxin. So whether oxygen will behave as a pranvayu or as a toxin, it depends on the metabolic pathways which microbes have mastered to gain the energy from the molecules. So the half of the planet age, there was no unbound molecular oxygen in the atmosphere. O2 is an unbound molecular oxygen, or scientifically diatomic oxygen. It's not oxygen, it is diatomic oxygen. NO2, unbound molecular oxygen, is an excretory product of the chloroplast. The plant cells also do cellular respiration and they also metabolize glucose. They need oxygen. But C6H12O6 plus O2 is a wrong beginning because students do not have an O2. And atmospheric O2 is secreted by plants. If O2 is usable in cellular respiration, why would plants throw it out? So let us understand also. This O is only present in the metallic world. There is nowhere in this planet you will find elemental oxygen. Now these are other forms of oxygen. Now you know O2, you know O3, you know triple O. I want to focus on the radical and the ion. Now, why I am touching BOD? Because atmospheric oxygen and aeration for water treatment, for successful sewage treatment, all that is claimed, and 70% of the energy expenditure in wastewater treatment is dedicated to aeration. BOD is a living cells organic polymer for growth and other cellular functions. Microbes living or a living system requires various organic intermediates like pyruvates, citrates, alpha-ketoglutarate, succinate. You all know all these members of the Krebs cycle. It is an intramolecular bond rearrangement within a molecule. BOD is an intramolecular bond rearrangement. We'll see what do I mean by that. A microbial ability to exploit the redox potential of an intermediate. That is the function of microbes utilizing bound oxygen. And BOD is of more significance to the food webs than the water quality. BOD has nothing to do with water quality. Either you accept it or correct. I am willing to learn, sir. Now, we are familiar with Krebs cycles, so I will only touch this stage. Citrate to cis-aconitate. This is the area of concern. I want to focus on one of this. Now, we all know glucose is converted to pyruvate, to citrate, isocitrate. Now, citrate, isocitrate, both are C6H8O7 molecules. Then what is the difference? Citrate loses one H2O. And this O comes from where? All textbook of engineering, they say that H2O is atmospheric oxygen. In mitochondria, where is the atmospheric oxygen? O2 going to enter. Now, what is the source of this H2O? Now, cis-aconitate citrate loses H2O and becomes C6H6O6, which is again, with the addition of that same H2O, is converted to isocitrate, which is the same molecular configuration. Rather, formula, not configuration. Now, what is citrate? Citrate is O6. I have shown it in a red color. Attached to the second carbon. And I have shown it in red, meaning it has higher ORP. You can go into that number wise, you can see how much millivolt energy will be released. That will be used for the synthesis of ATP. Now, isocitrate also has the same formula, but OH is transferred to the third carbon. And it is shown here in blue. This OH has a less redox potential. Citrate has a higher redox potential. Isocitrate. This. Now, the point is, we studied Krebs cycle from the human cell, liver cells, frog cell and all that, where the mitochondrion is dedicated to only produce energy. But prokaryotes, none of the prokaryote on the planet has all the enzymes to complete the Krebs cycle. Try to understand this point. Citrate to isocitrate, isocitrate to alpha-ketoglutarate, alpha-ketoglutarate to succinate, succinate to fumarate, malate, oxalate. Each step is influenced by one particular compound. Number two, not one single microbe can facilitate all the conversions. So it is a relay race. We all know that in a relay race, the first runner is focused on the gunshot. He has nothing to do with the finish line. and he takes a start before the others. The final runner is focused on the red ribbon. He should touch the red ribbon before any other competitors do. Now the mid runners have nothing to do with gunshot or the finish line. And that is the cycle. Entire STPs and activated sludge process, they are in denial of microbial cycle. So, following redox chemistry, high oxygen O2 is irrelevant. You know that here O2 is formed and again reutilized. And then we have all, because advanced oxidation is defined as microbial ability to utilize the OH redox potential. Now, after 100 years of ASP, activated sludge process was identified and introduced a century ago, but we have not moved forward. Now, reverse Krebs cycle, the master molecule is carbon dioxide, not oxygen. Because oxygen was not there for the first 2.3 billion years of the age of the planet. So, CO2 single carbon was converted to C3 molecule, which used proton, releasing 18 STP, and CO2 was produced. This cycle used to go on for first 2.3 billion years. This was the cycle. There was no glucose, there was no oxygen because there was no chloroplast, and glucogenic photosynthesis was nowhere on this planet. Why our textbooks start with C6, H12O6 is still something I have not understood. Now, glucogenic energy harvest, which is known as oxygenic photosynthesis, gave us C6. That is all we know. That C6 is converted to again. But C6 to pyruvate fermentation is a late introduction in the evolution of molecules and genomic machinery. Now, the fermentation, when all the four materials were all other conditions were highly unfavorable, microbes find out some way to get minimum energy to continue their survival. And those who could do that only can support glycolysis. Now, this glycolysis, C6, is converted to two molecules of pyruvate. That is again 18 HTP on the first left and 18 into 2 per pyruvate. The ATP gain is constant. Carbon dioxide kicked off the organic chapter of this book, starting with reverse. Now, this is what our textbook teach, and all I have read, whatever sewage treatment books I have read, they are working on this direction. This is all wrong. The starting point was this. Anyway, you all know this, so I will not. And carbon dioxide set the rules for the earthly planet, not glucose or oxygen. Now, the reverse Krebs cycle, highly specialized human body cells and all that, eukaryotic cells, they follow the conventional Krebs cycle, which is, you can say, like a clock. It goes clockwise. Some anti-clockwise reaction happens and succinate is converted to alpha glucan. Now, Dr. Nick Lane and his laboratory in Cambridge, they have found out a gene responsible for this reverse Krebs cycle mechanism, which leads to cancer. If you understand, if you must have read or listened to. There are so many YouTube releases also. So all the textbooks say that aerobic is more energetic, energy releasing than anaerobic and this and that. This is the terminal electron acceptor is a membranous domain. Nothing. It has nothing to do with the interior or exterior. Now we have substrate which has more 3 carboxylic acids. So that is why it is called tricarboxylic acid. And with 3 COOH, the acidic pH goes up, which would harm the cell. And H is thrown out, pH is restored, and the cell functions. Now this H is broken down into proton and electron. Now, terminal electron acceptor and proton mechanisms have separate origin and separate role. They came together, it's when the scientists are learning that. Now, energy gain of 18 ATP we say is common whether terminal acceptor is H2S, sulfur or oxygen. Now cytochrome oxidase came long before, rather 2 billion years before atmospheric oxygen. Anyway, we'll go a little far. Now, people in sewage treatment use facultative. Aeration दे दिया तो facultative हो जाएगा. Anaerobic will, that's not true. It's not science. The acetate switch turnover takes 92 minutes of quiescence. Otherwise, and then it's an irreversible switch. After you switch off the air compressor, it will not become anaerobic. Now, experiments with carbon. Should I go or should I skip Snail?
It's okay, you can go for some little more time, you can go.
Okay.
5 minutes.
So, 14 labeled carbon, glucose, we have CO2 is red, which is labeled. If you have methane, that is also labeled. So you can say that glucose is broken down to CH4 and CO2. But in normal cycle, this is apoptosis or breakdown. Pyruvate enters Krebs cycle through acetyl coenzyme A, and then whatever molecules synthesized, they carry labeled carbon. And the Krebs cycle, releasing carbon is black carbon, normal carbon. So Krebs cycle, at the end of that, we have the same carbon which was there with glucose, is a flawed inference. Now coming to this same picture, Dr. Rachel N. Dunlop, she is an Australian scientist. Then she switched over to Canada and then USA. All MND and ALS disorders, which are neurodegenerative conditions, are attributed to BMAA. And this is BMAA cheats serum. And it cheats even choline esterase. And the entire neuromotor mechanism is compromised. The same white man you had seen was a perfect young man. He was a cheerleader of the boat race competition. He is the same gentleman who suffered with this. Now, water soil magic. Now we go back to Dr. Snell's. Now, remediation of aquatic bodies begins with understanding zero order streams, which is nature's nano reactor. One first raindrop enters the soil. It's the micro reactor. The soil becomes moist, and there is a small microclimate moisture created. And the soil here, it settles down, becomes heavier because of the moisture. Now, this is an unparalleled magic. No other planets have that. Now, when the moisture is excess, it flows out, it forms streams, ponds, floodplains, lakes, rivers, and ocean. Now, oceans give you transcontinental atmospheric rivers. Shankar Mahadev was the atmospheric scientist of highest level. He understood cyanotoxins. He took the poison at the end of Samudra Manthan, but he retained it in the neck, so he is called Nilkant. So blue, green algae, Nilkant, these are all, if you see the synthesis between our earlier history, stories or whatever you want. And atmospheric river, it was so powerful that ordinary land could not hold it. Now the fresh water goes up, clouds, rains, but with heavy atmospheric moisture we have derived. Now, on the extreme side, we have all our land surfaces covered with impervious structures, concrete, houses, industries, roads, flyovers, and therefore, whatever the rain waters come, they ultimately run down. The soil is there. So the entire soil microbial gene library is finished. 1 gram of soil has a microbial gene library. If the DNA is unfolded, it will run into 1563 kilometers. Same thing, I am taking it to a little higher enlargement. So this is the earlier micro reactor. The raindrop with atmospheric CO2 becomes H2CO3, slightly acidic, which releases calcium and magnesium from the salts in the soil. And entire lush green, if it's the slightest rain, within 1 week we have all the soil covered with green grass and all the landscape. Now, wetlands are homeostatic. It's not just beautiful. I have enlarged the same thing here. Floating vegetation with all root system. And they have rhizome, which is called blue carbon. Why blue carbon? Because animal tissue engages 16 nitrogen molecules to 1 sulfur. S to N ratio is 1 to 6. In rhizome, it is 1 to 37. And therefore, denitrification is so efficient, and nitrogen, sequestered nitrogen is harmless. That is why we have 79% nitrogen in the atmosphere. We have emerging this thing and submerged also. Now, biotechnology, as I understand, is technology guided by bio. Technology is a tool to look deeper. Now this is no mixed eco-syndrome. I have done this work and all that. Now, we all know that in nature, urine and excreta are separate tubes. Why did engineers brought it together? I don't know. It was a single unfortunate economic decision to push both the waste through a common plant. Now, look, urine and excreta, what are the characters? Nitrogen dominant inorganic compound and excreta is carbon dominant. Both have different nature. Their decaying rates are different, and the microbes involved in the breakdown of urine and they are different. They have different pH demand, they have different multiplication rate, they take different time to work on each molecule of their concern. So I use this twice. Urine passage piping is different, and each has all potassium, sulfur and micronutrient, but occasionally. Excreta is loaded with pathogens. And STP sludge, that government municipal quarters, STPs, they sell it as a food manure, is a myth. There is no element of truth in that. This is a Western economics. Right? Now, food is equal to feces in nutrient equation. Once a human body is fully formed, let's suppose I take 100 units of nutrients. It goes down the gut. My body accepts 60 units because my body has metabolized 60. Out of 100, 40 units come from the gut straight away, but 60 come from the metabolic excretion. The total is 100. Now, unfortunately, sewage scientists, they do not link with agriculture. And agriculture curricula do not cover sewage skills. But well, this is something we'll discuss some other time. Detritus and sewage, carbon dioxide, they are heavenly. If there is no agriculture, there is no sewage. If there is no sewage, there is no agriculture. And mining finite phosphate rock is not sustainable. Well, President which is serendipity. Climate change is not new. It has happened earlier. Pandemics are new. It is only after urban development started that pandemics have started. Okay, we all manufacture weapons of mass destruction. How? Let us see. By default, we manufacture, because whatever we eat, carbon, hydrogen, all that, they are all converted to short-chain fatty acids. What we eat is apple, almonds, fruits, vitamins, everything. But what the body throws out, no book, no textbook ever shows that. Pathology is concerned with more of the nutrition and pathogens, medical science. But they don't talk about what throws out in a healthy body. Nor do the microbiologist studies. ये मेरा सिंह साहब मेरी एक शिकायत है आप लोगों से कि हेल्दी स्टूल की एनालिसिस क्या है, हेल्दी ब्लड की एनालिसिस का पैरामीटर्स है. Now all short chain fatty acids are C6 and below carbon number. You see, prokaryotes do not know glucose. Try to understand this. 6 and below carbon, they are short chain fatty acids. 12 to 6, they are mid chain, and 12 or higher up, they are long chain. Some other time we'll go into that. Now, nitrogen containing molecules, proteins, vitamins, DNA, and all that, they will form ammonia. Ammonia will be degraded to nitrogen. Now, both are strong greenhouse gases. CH4 is 30 times, N2O is 230 times. Now, CH4 has to be sequestered. That is through methanotrope. Methanotrope are in the soil. Now, nitrous oxide has to be sequestered to nitrogen through fungi, denitrifiers. Now, unless we find out a mechanism to balance CO2, CH4 and N2O cycle, there is nothing we can do as far as the climate change is concerned. Now,
Muni bhai, in 2 minutes we will now
Haji,
wind up.
okay, I'll jump on to something more important. Yes, nature does not reinvent. I was taking this class, and by explaining sewage treatment, I put a colon picture on my body. Each region has different microbial population, communities, pH conditions, raw material is separate. It's a very complex ecology. And synergy of the highest order, STPs can never meet it. So quiescence, anoxic zone, colonic fermentation, and water and nutrient recovery is the function of colon. Why did we put so much of water? Just for pumping convenience? That's not engineering. Now, be it a river or human body, esophagus, from mouth to esophagus, there is no dilatation. It's a straight flow as a river flows down the river. High velocity zone, but colon is the estuarian. Mangroves, low velocity, high duration. Now, if you see the colon microbial population, Firmicutes, then Bacteroidetes, they are there in the colon, they are there in the estuary. With some percentage of community may change, but they are there. They are there in the paddy, they are there everywhere there are water bodies stagnant. But STPs have 8 to 10 times more nitrous oxide compared to other these ecological conditions, because these denitrifiers do not have enough time to work on nitrous oxide. Look, nitrous oxide on the column, I had posted it in my our WhatsApp group also, the nitrous oxide, where it is formed. So nature does not reinvent design procedures. Reverse or colon, organic degradation follows the second law of thermodynamics. I think these are the microbial analysis. I will not thank you.